Hello, everyone. This is Thomas Sir. I'm the CEO of Kinexum and EVP of the not-for-profit Catalyst Institute, the co-sponsors of today's webinar, Mitochondria, Aging and Health. Today's webinar is an update and an expansion of a session that we held at our virtual Metabobicity conference this past October. Just earlier this week, the journal Cell published an extensive piece co-authored by Metabobicity speaker Guido Kromer, suggesting that the mitochondrial function and dynamics seem to play a leading role in each of the hallmarks of aging as defined by the authors. So I'm really looking forward to our expert panel discussion. A couple housekeeping points before we begin. One, we are muting folks as they come in to preserve the sound quality. Two, and if you notice that you are live and you're not a panelist, please put yourself on mute so we don't hear dogs barking and so forth. Second, because of the large number of attendees, we can't really do a radio call-in show format, at least during the formal part of the presentation, recognizing members who may have a question. So rather, if you have a question along the way, please type it into the chat box column on the right, and the panelists will try to answer as many as they have time for. Three, we are recording this webinar. So a video of this panel presentation and the Q&A and a PDF version of the slides will be made available on our Kinexum website or Catalyst website, I think, within a couple business days following today's event. All registrants will receive links by email when these materials are available. And finally, just to close with a commercial for our next Kinexum webinar on January 29, entitled Wow or Yow, FDA Outlook for 2021 and Beyond. And in February, Stanford geroscientist Tom Rando and Harvard evolutionary anthropologist Dan Lieberman will present on the molecular and evolutionary bases for exercise as a geroprotector. Now, I'll introduce the moderator shortly, but first I want to introduce Adrian Berg, Executive Director of our not-for-profit Catalyst Institute, which was set up last year to start taking on the responsibility for our Metabobicity conferences and webinars like this one today. Adrian, please take the mic.
So thank you so much, Tom. I'm very, very delighted to be here today, and I want to welcome all of you who are on this webinar to be part of the new Catalyst Institute. We have a very strong mission. We call it the Moonshot, and what we want to do is accelerate the really wonderful geroscience breakthroughs in aging so that they become translated into real material and sustainable public health policies. We don't have a national public health policy on health span or on longevity. And as we heard from Thomas a minute ago, we have a lot of responsibility. One of them is to do our wonderful annual conference. It's going to be for the 4th time, either hybrid in Washington, D.C. and virtual or just virtual. That we're not in control of. But because our mission has so much to do with healthy longevity, we might be the ones in control of future pandemics. We're here to strengthen the host. And I would like all the geroscientists and like myself, I'm not a scientist, I'm a lawyer, but don't hold it against me. I am very, very much involved with longevity. I'd like you to be as well. Now, the Institute is brand new. It got started in April in Virginia. We are now getting our status as official 501c3 corporation. And it was the inspiration of Zan Fleming. Now, so are these conferences, and it builds on the entire Kinexum team and their camaraderie in making sure that we can take that moonshot to healthy longevity. We're going to have the conference. I would like you to know all about it. And we have other activities like Project Health Span, where we're looking to see if we can create a national policy on longevity, as they have in the UK, and Project Endpoint, where we are going to see whether we can get regulations with regard to longevity a little bit safer and a little bit faster to the people. So just watch for the launch of a brand new online community called the Catalyst Institute Campus. You'll find that on our website, which we're just building, catalyst.org, and we will be able to become a community. And what we ask of you is that you join, you participate, build forums, talk with each other, and take the moonshot with us. Thanks, Thomas. So now on with the show.
Thank you, Adrian. Now to our expert panel. You can read their bios in the publicity for this webinar. But it's an honor and pleasure for me to hand the mic off to noted mitochondrial population geneticist or genetics expert, Dr. Doug Wallace of CHOP, the Children's Hospital of Philadelphia, who among many other things, pioneered the theory of mitochondrial Eve, who will introduce the rest of the panel. So Doug, please take on the mic.
Yes, thank you very much. It's really a pleasure to be here and to thank Connexin and Catalysis for their developing this program and letting us have it with inviting all of you. Just a minute, I'm working on getting you out of the way here. Very good. Go back one slide, please. So these are our panelists. I'm really pleased to introduce Francesca Fieni. She is the founder and chief scientific officer of Pano Therapeutics, and we're going to hear from her about her important work in mitochondrial channels and metformin. And Hazel Cizico, who is the director of research and social profit network, excuse me. And Hazel pioneered in developing a pentapeptide that has been very, very important in the development and maintenance of quality of mitochondrial intermembrane. And Michael Zemel, founder and chief scientific officer of NuCert Biopharmacology, and he is a leader in how we might use nutrition to maintain the quality of our life. Can we go on to the next slide, please? So really want to thank Connexin for giving us this opportunity to speak with all of you. The topics we're going to have in order, I'm going to give a slight overview just to get us in frame. My topic will be mitochondrial etiology of metabolic and degenerative diseases. Then Hazel will talk about repairing mitochondrial structure to restore ATP function, and Francesca on addressing complex diseases of aging by targeting mitochondrial ion channels, insights into metformin's action, and then Michael on mitochondrial nutrient sensing as a target for therapeutic intervention. And I really want to thank the people in Connexin who have been really helpful in getting this together. Bonet Jenkins and Elijah Abbas, Zan Fleming, and of course, you've just heard from Adrian Berg. So they've made all this possible. Next slide. Okay, so the mitochondria is a component of our cells, of course, but what's really interesting is that it's a separate life form. The two life forms came together about 2.5 billion years ago to form a symbiosis. One was an archaeobacteria, and that ultimately gave rise to the nuclear cytosol, and the other was a oxidative bacteria, and that gave rise to the mitochondria. They then began to exchange genetic information and become specialized, and ultimately the nuclear cytosol became specialized in encoding information related to structures and ultimately to development and then complex organisms, whereas the mitochondrial genome became increasingly smaller and it became specialized in generating energy. And so the mitochondria then is the power plant of the cell. And the reason the genome had to get smaller is because each bacterium, if it has a free living lifestyle, it needs to use most of its energy to replicate its DNA, transcribe it into RNA, and translate it into protein. So if all of the mitochondria inside the cell had their own normal sized genomes, they would use up all the energy and there would be no advantage to the nuclear cytosol. But what happened is that genes were transferred, all the genes for structure were transferred from the mitochondrial DNA to the nuclear DNA chromosomes. So instead of having hundreds of copies, one in each mitochondrion, you now only needed 2 copies of each gene. And that then cut down the amount of proteins that needed to be made in the mitochondria and gave you about 100 to 1,000-fold saving of energy. And all that extra energy then became available to build complex structures, encode large numbers of genes in the nuclear genome, and then to create then higher organisms. What's interesting is in the process of transferring those genes to the nucleus, the mitochondrial origin genes were basically transferred randomly into the nuclear genome. And so one of the most interesting aspects of this problem now is how do you regulate 1 to 2,000 nuclear genes that are all required in stoichiometric levels to assemble the hundreds to thousands of mitochondria within the cell. And that then links together then the interaction of the energetics of the mitochondria with the epigenome of the nucleus, and then the whole issue of how aging occurs through both changes in the nucleus and in the mitochondria. Next slide. So the mitochondria generates energy, according to Peter Mitchell, by what's called a chemoosmotic coupling system. And that's basically by which the mitochondria generates an electrochemical gradient. Now, the mitochondrial DNA codes for 13 polypeptides, 7 of which are subunits of the first enzyme of mitochondrial function, that is enzyme NADH dehydrogenase, and that has 45 polypeptides, of which 13 are coded in the mitochondrial DNA. The mitochondria codes for cytochrome B, which is one of the 11 polypeptides of respiratory complex 3, 3 polypeptides of the 13 polypeptides of respiratory complex 4, cytochrome C oxidase, and 2 polypeptides of the last enzyme of the oxidative phosphorylation, the ATP synthase, which has about 18 polypeptides, 2 from the mitochondrial DNA. And these polypeptides are encoded in the mitochondrial DNA, transcribed into RNA, and translated on mitochondrial ribosomes, which are sensitive to inhibitors such as chloramphenicol and aminoglycosides, and they assemble then the polypeptides inside the mitochondria. Now, what's interesting is if you look on the left, the structure of the mitochondrion shows that they're infolded mitochondrial membranes, and these are known as cristae. And this is a tomogram, you can then go ahead and get it started, of the mitochondria. And what you can see, as hopefully we can move through the mitochondria over time, is that these infolded cristae are aligned across the mitochondria, from one mitochondria to the next. And so clearly the mitochondria are interacting with each other and communicating. And so we've been very interested in how that happens, because the enzymes, complex 1, 2, 3, 4, and 5, are all arrayed along those infolded membranes, the cristae. So if the mitochondrial electron transport chain, complexes 1, 2, 3, 4, and 5, pump protons into the lumen of those very thin cristae lumen, then we have high proton gradient in a very, very small volume, and that's shown in the middle lower structure. And there are particular proteins such as OPA1 and the mucose complex that actually close off the outer ends of those cristae so that there's a concentration of those protons, creating a very, very high electrostatic potential. And then the ATP synthase is using that electrostatic potential. The protons flow through the ATP synthase, and that then drives the condensation of ADP and phosphate to ATP. And that then exchanged across the inner membrane by the adenine nucleotide translocators. So if you look at the upper right-hand panel, what we really have then are these cristae lumen with very high positive charge, and we think that those are electrostatically repelling each other across the membranes of the different mitochondria, which are linked together by intermembrane junctions. And that then couples the electrostatic potential across long arrays of mitochondria. Then what's shown in the lower right-hand corner is the structure of the ATP synthases, which are always dimers, and they're always arrayed along the outer edges of the cristae. That's where the electrostatic potential would be greatest. So the whole thing is designed around basically developing a very strong biophysical system for driving energetic potential. And if you break the OPA1 and open those red dots there that form the closure of the cristae lumen, then your efficiency drops off dramatically, and ultimately you mitochondrial intermembrane cristae swell, and that can lead to something called the intrinsic pathway of apoptosis. Next slide. Go ahead again, one more, please. Okay, so this is a classic example of an inherited mitochondrial DNA disease. Oops, back one. And the mitochondrial DNA is unique and it's transmitted only from the mother to all of her children and from her daughters to their children, whereas male's mitochondrial DNAs enter the egg and are seen as foreign and are destroyed. So here we have a situation where we have a mutation in the mitochondrial DNA that is heteroplasmic, that is a mixture of mutant and normal mitochondrial DNAs. And this particular mutation in the ATPase6 changes the leucine at codon 156 to an arginine. Now, this is a family we studied in the 1980s, and the boys at the lower right-hand corner in red, they either had cerebellar ataxia or they died of a lethal childhood disease called Lee syndrome. Their mother had two different husbands that gave rise to the different children, and she had retinitis pigmentosa, and her mother's mother had retinitis pigmentosa, had two consorts, and all of her daughters also had retinitis pigmentosa. And the mother's brother, up on the upper left-hand corner, he had this massive neurological disease shown in the middle, where the cerebellum is now about half of its volume and the brainstem is about half, and that's olivopontocerebellar atrophy. So the children at 1, 2, and 3 in the lower lineage 4, they then developed Lee syndrome, and that gives a bilateral striatal necrosis, which is shown on the far right, those leaf shape patterns inside the center of the brain. So what's amazing about this mutation is that it's heteroplasmic, and the individuals that died of childhood disease had about 80 to 90% or 100% mutant. Those with retinitis pigmentosa had 75% of the mitochondrial DNA mutant, and the individual with the severe neurological disease had between that 80 to 90% of the mutant mitochondrial DNA. So subtle changes in heteroplasmy can have a huge effect on the clinical phenotype. And the way this works is, if you look at the lower right-hand corner, we have the ATPase6 gene, which is mitochondrial DNA coded, and the ATPase9 gene, which is nuclear encoded. And the ATPase9 gene has a glutamate in the middle, that's GLU, and that glutamate is negatively charged. So the proton can go from the lumen of the Christie to the glutamate and then on into the matrix. And it's allowed to do that by a leucine on the left-hand side that forms a hydrophobic pocket. But this mutation changes the leucine to an arginine, which is positively charged, and that reacts then and forms a salt bridge with the glutamate, and thus locks the synthesis of the ATP synthase. So you have a direct quantitative effect on ATP based on the percentage of mutant mitochondrial DNAs. Next slide. Now, the mitochondrial DNA being maternally inherited, it cannot recombine because it never mixes male and female mitochondrial DNAs. So what that allows you to do is, by sequencing the mitochondrial DNAs of any two people in the audience here, the number of mutational differences is directly proportional to the time you shared a common mother. So what we were able to do then is analyze the mitochondrial DNAs from populations around the world and basically reconstruct human migration. And so if you look at Africa in the upper left-hand column or picture, you see that L0 are the original mitochondrial DNAs, and those are found in the Khoisan people of the Kalahari. L1 and L2 are pygmy mitochondrial DNAs, one at 90,000 years old and the other at about 30,000 years old, two different origins. L3 are sub-Saharan Africans, and then in Ethiopia, two mitochondrial DNAs arose, M and N, and only these mitochondrial DNAs left Africa and colonized the rest of the world. So M stayed in the tropics all the way down to Australia, and then much later moved into the temperate zone to colonize the Asian populations. But N moved directly into the temperate zone, gave rise to all of the European mitochondrial DNAs, and then moved laterally to give rise to half of the Asian mitochondrial DNAs. And then those mitochondrial DNAs that became adapted up in Chukotka in Siberia on the far right-hand side of Siberia, were in a position then about 20,000 years ago to cross the Bering Land Bridge and colonize the Americas. So the reason that this high correlation of mitochondrial variation and population diversity occurs is because the mitochondrial DNA variation of populations is functionally relevant, and it changes mitochondrial function to allow our ancestors to adapt to different environments. So these variant mitochondrial DNAs then are also metabolic risk factors for a whole range of common diseases. So if we look at the lower left-hand panel, we can see that lineage D5, F4, and N9A under level 1 lineages, those are protective of diabetes. Lineages F4 have increased risk for hypertension, and lineage F4 is also at high risk for obesity. We can also look at European lineages and look at something like neurological disease, in this case, autism. And we can see that European lineages I, J, K, T, U, and X all have about a odds ratio of 2 increased risk of autism. And that's interesting because they represent 55% of the European population. So there's a huge mitochondrial component to risk factors for autism. And then finally, a number of groups have looked at mitochondrial DNA variation in relation to longevity. And among Europeans, lineage J and lineage U are very important in increased longevity. And similar finding is for lineage D in Asians. Next slide. So basically then the other thing that happens is as we age, the thousands of mitochondrial DNAs in our cells accumulate mutations, and they then erode mitochondrial bioenergetics. So on the upper left, we have basically 2 graphs. One that on the left shows the, say, representation of the amount of energy output. And then over time, as mitochondrial DNA variation accumulates, we see an erosion of energy. And then different organs have different reliance on mitochondrial energy. The brain, being 2% of our body weight, uses 20% of all of our mitochondrial energy. So as our energetics declines, the first thing that is affected is the brain, followed by the heart, the muscle, the renal, and the endocrine system. And those are all the organs that are affected with aging. And to prove that, we made a mouse in which we took catalase, which is normally found in the peroxisome, and we took off the peroxisomal targeting signal and put on a mitochondrial targeting signal shown in yellow up on the upper right-hand corner, and made a transgenic mouse. And when we express that, catalase in the mitochondria, we remove all the mitochondrial hydrogen peroxide, the antioxidant defense, it's an antioxidant defense. And what that does is it increases the lifespan of the mice by 20%. And so what you can see on the lower left-hand corner is a young and old mouse, whereas young mouse, the mitochondrial DNA is full length, whereas in the old mouse, we see accumulation of mitochondrial DNA deletions. And we see the same in humans, young and old, on the middle graph. And then on the right, what you can see is the mice with the high catalase in the mitochondria have about a 50% reduction in mitochondrial DNA damage. So thus we've linked then longevity with mitochondrial DNA decline. Next slide. So basically then in summary, one possibility is that if we put energetics in the middle of human biology, then oxidative phosphorylation can be affected by changes in the nuclear genome, by mutations or changes in the epigenome. It can be affected by mitochondrial variation, either in maternally inherited mutations or ancient polymorphisms. It can be affected by the diet we have, or whether we exercise or are exposed to toxins. And if you inhibit mitochondrial function, even partially, the first things that are going to be affected are the most energetically demanding organs. That's the brain, the heart, the muscle, and the renal systems. And those are all the ones that are first affected in aging. Also, if you inhibit mitochondrial function, you'll not be burning carbohydrates and fats, which are, of course, the substrates for oxidative phosphorylation, and they will accumulate. And then that's what we call metabolic syndrome, giving rise to cardiovascular disease and hypertension. And the mitochondria is a perfect bacterium. So if the energetics is affected, then apoptosis fails and the mitochondria are released in the bloodstream, and that activates then the whole inflammation process, the C-gas sting and the inflammasome pathways. And that's why all the common diseases have an inflammation component. And finally, as aging continues and energetics declines, ultimately all these organ systems begin to fail. And we believe that's an important component of aging. And finally, cancer is all about having enough energy for the cancer cell to grow. So with that, hopefully brief background, we can now hear from the really important contributions of our colleagues.
I hope you can all hear me. thank you for this opportunity to speak here. And Doug, thank you for the marvelous introduction, because you've just about covered everything that I'm going to talk about in my next 10 minutes. Next slide, please. So, Doug introduced the idea of this endosymbiosis theory that Lynn Margulis published in 1967. It is the evolution of mitochondria inside our cells that allow us this energetic boost that was necessary to support the building and the maintenance of complex cells. So I will just focus a little bit more on the ATP-dependent cellular processes inside our cells. Doug already mentioned that the biosynthesis of macromolecules already consume about 2/3 of the ATP inside our cells. And that is especially in terms of lipids and proteins. So you can easily imagine then ATP deficiency will inhibit cell proliferation and differentiation. But ATP is also necessary to maintain the cytoskeleton of cells, to support all active transport processes in the cell, and even for cell signaling, both ATP as its own as a signaling molecule and cyclic AMP for all the GPCR pathways. And perhaps less well appreciated is the fact that a lot of ATP is required for protein quality control. Protein folding has a high energy barrier, and even protein degradation by either the ubiquitin proteasomal system or the lysosomal system requires ATP. And lastly, of course, ATP is required also for apoptosis and autophagy if one is to die by a sort of cell suicide rather than necrosis and inflammation. And there are, of course, specialized cell functions such as synaptic transmission and muscle contraction that would require even more ATP. So next slide, please. So it's been estimated that ATP turnover rate is as high as 65 kilograms per day at rest, and it can go up to even 200 kilograms a day. And here is the graph. Doug already mentioned the tissues that have the highest ATP demand. So how can mitochondria possibly produce this much ATP per day in the human body? 65 kilograms. So here I will say that this is due to a very unique and beautiful structure of mitochondria. Mitochondria are made up of the two membranes, the outer membrane in dark blue, the inner membrane in light blue, and then the cristae membranes, which are invaginations from the inner membrane in yellow. You can see that the densely packed cristae membrane shown here will greatly increase the surface area to accommodate all the protein machineries that is necessary for the electron transport chain shown below. And these curvatures, they're so tight, they're less than 10 nanometers. What makes it possible is the existence of cardiolipin, shown in red in that schematic. And cardiolipin is a unique phospholipid that is of bacterial origin, and it has a conical structure, so it helps bend the membranes. Next slide, please. Next slide, please. So while cardiolipin is really important for bioenergetics when it is on the inner membrane, it is also in an area where the electron chain can leak electrons and cause the production of reactive oxygen species, and that will cause lipid peroxidation. When that happens, when cardiolipin is peroxidized, it will degrade, and so you will have a degradation of the cristae membranes. And on top of it, oxidized cardiolipin will translocate from the inner membrane to the outer mitochondrial membrane. So the first thing that happens is that with the loss of the cristae membranes, you have less ATP production. But beyond that, the cardiolipin sitting on the outer membrane will represent a danger molecule to the rest of the cell. So what happens now is this cardiolipin can interact with ER membranes and start the process of mitophagy and even autophagy. We know that oxidized cardiolipin will also cause the release of cytochrome C, which then leads to the initiation of the apoptosome to mediate apoptosis. And finally, more recently, it's been greatly appreciated that oxidized cardiolipin will form a docking station for NLRP3, ASC, and IL-8 and caspase-1 to initiate inflammasome activation. And inflammasome activation is now appreciated to take place in aging, which is also called inflammaging, and to be the underlie the pathology of many age-related diseases. Next slide, please. So here are just some examples to show you how Christie membranes can be lost in aging. On the left, transmission EM taken from young mice, cardiac and retina mitochondria from young mice, and you can see a lot of dense Christie membranes. However, when mice get to be about 2 years old, you will see that there's now no clear definition of Christie membranes in many of the mitochondria, not all, but many. And yet the inner mitochondria membrane remains intact, but it's just a loss of Christie membranes. The same is actually seen in heart failure. This is cardiac mitochondria from a human with heart failure, and you see the loss of Christie membrane. And it has now been shown that this is associated with loss of cardiolipin content. And finally, you see examples from Alzheimer's disease and Parkinson's disease with loss of Christie membranes, but intact inner mitochondria membrane. Next slide, please. So these studies would suggest that it is really important to try to repair cardiolipin content and restore these Christie membranes if we are to restore bioenergetics. So we've been very fortunate. We discovered a series of small peptides. They've been called the cetoschiller peptides. And SS31 is probably the most studied example of them all. SS31 was shown to specifically target to the inner mitochondrial membrane, bind to cardiolipin, and prevent the peroxidation of cardiolipin. So you will see that this would now prevent the collapse of the Christie membranes. And there are studies that have shown that treatment with SS31 can prevent this cardiolipin oxidation and loss of Christie membranes. But there are even studies to show that we can even repair the loss of cardiolipin in aging animals and restore the normal mitochondria architecture. Next slide, please. So here's just the examples. On the left side is pre-treatment, prophylactic protection of kidney mitochondria in ischemia, which is shown that you have loss of Christie membranes, and treatment with SS31 preserves them. In a model of type 2 diabetes that is caused by high-fat diet, there's also a loss of Christie membranes in kidney tubular mitochondria, and they are preserved if the SS31 is given. Interestingly, this is even when SS31 does not alter blood glucose levels. And then on the right side are the aging studies where old animals show this loss of Christie membranes, and 2 months treatment of SS31 started in late life can now restore Christie architecture. Next slide, please. And so if you follow this schematic and this understanding, then treatment with SS31 should be able to prevent all this downstream cascade of events that can lead to aging as well as age-related pathologies. Next slide, please. So I will simply summarize quickly what all the studies have shown. There are numerous studies by many investigators where with the treatment of SS31, in animal studies and in humans, that we're able to optimize ATP production and reduce ROS production. And this is ROS production. This is simply by promoting electron transfer. And in doing so, SS31 has been shown to inhibit all types of cell death in stress-mediated models, such as ischemia. It is also shown to attenuate inflammation by downregulating any release of NF-kappa B, TNF-alpha, and the interleukins. In aging animals, they've been shown to reduce senescent cells. In tissue damage, they can promote cellular repair and accelerate the regeneration of kidney cells, for example. They've also been shown to promote cellular metabolism by upregulating SIRT1 and AMPK kinase. They can improve organ function, and in vivo efficacy has been shown in numerous age-related chronic diseases. So finally, I will simply come back to what Doug ended up saying, is that mitochondria dysfunction may well be the centerpiece in the hallmarks of aging, because clearly with insufficient energy, one can expect altered cell-cell interaction. You can expect loss of proteostasis, induction of cell cycle arrest and senescence, exhaustion of stem cells. So this is where, I think that I would go along with many of the authors in the paper that Thomas mentioned at the beginning, that we think mitochondrial dysfunction lies, center amongst all the hallmarks of aging. Thank you.
Hi guys, thank you very much. Hazel, great talk, of course, and Doug, the same. Thank you, Kinexon, for giving us the opportunity to redo this session that was kind of lost during the Metabizity event, so that's great. So I usually like to point out that I'm not a gerontologist. I never work in biology of aging, so I just happened to cross path with the space due to a discovery that I made while I was at UCSF, which basically launched this company. So Pano Therapeutics is a UCSF spin-off that has the overarching goal of addressing complex diseases of aging by targeting mitochondrial ion channels. So why do we care about mitochondrial ion channels? Next slide, please. So as already Doug and Hazel went through, clearly there's agreement that bioenergetic decline is at the root of aging, and the two pillars of healthy aging, which is physical activity and a healthy diet, they affect bioenergetic metabolism, so mitochondria must be very relevant players in the game of healthy aging. Next slide, please. So what is bioenergetics? From a mere biophysical definition and perspective, it can be defined very simplistically as the buildup of an electrochemical gradient across the inner mitochondrial membrane. And this electrochemical gradient, which is also known as the protomotive force, and for which Peter Mitchell won the Nobel Prize in 1978, is composed of both a proton gradient and then electrical potential, which is the infamous mitochondrial membrane potential. So this electrochemical gradient is coupled to phosphorylation of ADP into ATP. So basically, any positive current that crosses the inner mitochondrial membrane can actually uncouple the phosphorylation of ADP from this electrochemical gradient. As you can see here, an uncoupling current can be mediated by protons, can be mediated by monovalent cations like potassium and sodium, it can be mediated by calcium. So next slide, please. So there's many ways of basically uncoupling mitochondria, and usually we forget about all of the others because we focus on only the ones that we know the most. So there's the famous DNP, which is a proton of force that basically shuttles protons across the inner mitochondrial membrane. There's fatty acid mediated kind of uncoupling, which is mediated by proteins of the likes of UCP1 in brown fat of mice. Humans don't have brown fat, so most likely this fatty acid mediated uncoupling in humans is mediated by ANT in skeletal muscle, heart, and liver, which is also shuttles, of course, protons across the inner mitochondrial membrane. And then there's kind of like a long time forgotten way to uncouple mitochondria, which is through proton ionophore of the likes of valinomycin, which basically carries potassium across the inner mitochondrial membrane, specifically potassium. So any basically potassium channel or transporter that shuttles potassium across the inner mitochondrial membrane has the potential to uncouple mitochondria. So how is uncoupling relevant to physiology and pathophysiology? Next slide, please. So what's important to know is that, first of all, uncoupling is never intended to be switched on all the time. It is basically supposed to serve a physiological function where it is open up only in brief periods of activation when the mitochondrial membrane potential is hyperpolarized. There's a lot of work by Guido Kromer about that back in the 90s. So when this happens, and the reason the mitochondria do that, they mildly uncouple the mitochondrial membrane, is to basically protect the cells from acute exposure to reactive oxygen species. However, and unfortunately, if this mild uncoupling keeps being activated, so our cells are chronically exposed to mild uncoupling over time, that basically that chronic activation of mild uncoupling enables ROS, reactive oxygen species, to be maintained at the submaximal threshold that does not trigger necessarily mitophagy or all the good things, but they start to become signaling molecules of death. They become mutagens, they're signaling molecules for uncontrolled cell proliferation, they're oncogene stabilizer, and so on. So at that point, mild uncoupling from being a good thing becomes a very bad thing. Next slide, please. Next slide, okay. So as I was mentioning before, I'm just gonna focus on the protein-mediated type of uncoupling, not the protonophore ones, which are really basically more aggressive wave uncouples the mitochondria. So there's a very well-known ones, which it's basically a thermoregulatory uncoupling. Again, is mediated by thermogenin or UCP1 in brown fat and ANT in skeletal muscle and heart. It is free fatty acid mediated. It's non-omic, and the ions transporters are protons, and it modulates mostly the proton gradients in the protomotive force. And then the overall effect of this is affecting body mass of the animals. As I mentioned before, there's other forms of uncoupling, and there's still a lot of questions around the molecular identity of this type of uncoupling. It is non-thermoregulatory. It's expressed across many tissues, skeletal muscle, heart, proliferating tissues all over the place. It is non-free fatty acid uncouple. It is non-fatty acid dependent, sorry. It's a mild way of uncoupling the mitochondria. It is homic, and the ions transported are supposed to be monovalent cations of the likes of potassium and sodium. And thus, for this very reason, it's supposed to mostly modulate the mitochondrial member potential. I would like to point out that in bacteria, the protomotive force is mostly regulated by the proton gradients, whereas in mammalian mitochondria, 2/3 of the protomotive force are due to the mitochondrial member potential. So whichever ions, whichever cations modulates this very variable in the protomotive force is really important for the making of ATP. And of course, you know, this force of uncoupling would be associated to the modulation of reactive oxygen species, as I described before. Next slide, please. Which brings about the discovery that basically launched this company, which I made while I was at UCSF. So I am in electrophysiology by formation, and to the electrophysiologist's eye, the very properties of metformin, which is a monovalent, basically is an organic cation positively charged. It's very much looks like a typical inhibitors of potassium channels. So as you can see here, the power of electrophysiology applied to mitochondrial members is the ability to dissect different mechanisms that mediates for ion fluxes in the mitochondria. And so strikingly, to our surprise, while I was at UCSF and I was looking into a pharmacological profile of the potassium sodium current in the mitochondria, I found out that it's the target of metformin. So we have done a thorough biophysical and pharmacological profile. We're still gathering a lot of information and genetic information in collaboration with Doug Wallace to identify the subunits that are responsible for this mechanism in complex 1. Which of course I'm not going to go into details here. There's a patent going, and we're going to submit a publication, so everything is going to be out there. Next slide, please. So I wanted to pull these figures from one of the latest reviews by Nir Barzilai. As you can see here, there's many signaling pathway downstream of complex 1 that might be associated to complex 1 and explain the hallmark of healthy aging. Of course, everybody has their favorite mechanism of action. We favor the idea that you know, there's no direction of causation in this necessarily in this slide, but our preference is that actually complex 1 sits at the very front of many, not all, of course, of the mechanisms that are described here. And one of the main questions that is still lingering out there is if the clinically relevant concentration of metformin actually hit upon complex 1. So I wanted to clarify here that from a biophysical perspective, the amount of metformin that is supposed to reach the mitochondria it's easily explained, but what in biophysics is defined as the nurse potential. So basically, metformin is a positively charged cation. It's supposed to have two charges dislocated across the two guanidine moieties, but in the clinics is used as a chloride salt. So there's one positive charge. That basically means that any tissue or any cell that expresses a transporter of the likes of OCT1, which enables metformin to cross the membrane, allows metformin to accumulate in sub compartment of the cells based on the membrane potential. So mitochondria have a membrane potential in the likes of minus 180 millivolt to minus 200 millivolt, which basically translates for patients that are taking metformin at 1 grams per day. Their plasma concentration ranges between 10 to 100 micromolar. That basically, based on nurse and provided there is a transporter to get metformin in, translates into subcompartmentalization into mitochondria in the between the 40 to 80 millimolar range within the mitochondria. That's way above the IC50 of metformin for complex 1. So idea here is that a lot of people actually argue that when measuring the amount of metformin in the liver is basically the same as the plasma concentration. I would like to point out that that's not the way to know where metformin locates, because the volume of the liver is different. So this is just to trigger that because I know that it's a question out there. What is the most relevant target of metformin that would mediate for most of the biological effects of metformin? We favor this hypothesis. Of course, we welcome others, but we think that this is very important mechanism. I have 2 extra slides, but I don't think we need to go through them. Thank you for your time. And yeah, just here, 1 second. Yeah, the one before. What we're basically doing at Pano here, I have publication on different ion channels in the mitochondria. We're gonna, again, submit the paper for the potassium sodium current. We're gonna be developing drugs that are not gonna be necessarily at this stage used for preventing aging. We still don't have enough good endpoints to determine that. So at Pano, what we're doing, we're developing first and foremost drugs for pathological conditions of the likes of diabetes and cancer. Thank you very much. I hope you enjoy. And Michael, I'm going to give it to you.
That was a wonderful talk. As this is tough. I have to follow 3 really wonderful talks. So I'm going to switch gears a little bit and focus on a little more translational component, focusing on nutrient sensing or energy sensing as a target for therapeutic intervention. If we can go to the next slide, please. Just a quick overview. We're all familiar with the hallmarks of aging. They've been discussed many, many times. I show you this slide from a recent paper that represents it as a network model. And the only reason I'm showing you the slide is to point out that in the middle of the output side on canonical aging mechanisms sit both nutrient sensing and mitochondrial function. And I will be talking about the nexus of the two. I will point out that in the middle of this slide, I'm not presenting this from my computer, so I don't have a pointer, but in the middle of this slide, you will see 5 bubbles. Of those 5 bubbles, SERTs, IGF-1 insulin, mTOR, and AMPK, 4 of them are nutrient sensing or energy sensing systems. So let's move to the next slide. Mentioned hallmarks of aging. I want to switch for just a moment to hallmarks of health. This is a wonderful paper that was just out in the last few days in Cell, Hallmarks of Health. And again, if you look in the middle left lower, you'll see hormetic regulation. And that really is where I want to focus a discussion of nutrient sensing, really mitohormesis as it affects health span and lifespan. So let's move to the next slide. So key energy sensing targets. Well, certainly for sensing low energy, as during fasting, starvation, excessive exercise, et cetera, key molecules are the sirtuins and AMPK. And that is where I will be focusing our discussion. When I focus on sirtuins, I'm focusing primarily on SIRT1 and SIRT3, as they either indirectly or directly affect mitochondrial function. I'm acutely aware, of course, of the anabolic energy sensing shown in the lower right, where we're looking at the mechanistic target of rapamycin, and it of course interacts with both sirtuins, AMPK as well, but that will not be our focus today. Move to the next slide. The key focus of NuSirt has been on the amino acid leucine, and our key finding has been that leucine is an allosteric activator of SIRT1 and SIRT3. The figure only shows SIRT1, which basically causes a leftward shift in the NAD dose response curve, allowing activation at concentrations of NAD that are somewhere between 50 and 80% lower than would otherwise be possible. And this enables robust synergy with NAD donors, with sirtuin activators. Let's go ahead and go to the next slide. Thank you. So our overall framework is not that leucine is going to be a sufficient nutrient sensor by itself to have a therapeutic effect, but rather that leucine will co-activate other compounds. And in co-activating, it will amplify the effects of either sirtuin activators or downstream effectors of sirtuin activators. And like many, I have a theory of everything. And my theory of everything, of course, covers this range of diseases from mitochondrial dysfunction to cardiovascular disease. But I will limit my comments today to those that we have data for. Let's go to the next slide. Excuse me. So here, I'd like to spend a moment talking about leucine synergy with NAD donors. You may have seen a fair bit of literature on NAD precursors that can be taken orally, nicotinamide riboside, nicotinamide mononucleotide, as sources of NAD, which theoretically, as activators of SIRT1, which have downstream effects on mitochondrial function, should show some of the same systemic benefits shown on this slide. With multiple clinical trials of nicotinamide riboside in place now, I think we can safely say we do not generally see the benefits that are seen in non-clinical models. What we can say is that these compounds are safe and that they somewhat increase NAD, perhaps twofold. That is sufficient to, that is simply not sufficient or appears not to be sufficient to have glucoregulatory effects, lipid oxidation effects, or really effects on any measurable disease state. So our approach has been a little different. NAD supplements, or nicotinamide riboside and nicotinamide mononucleotide supplements, can basically increase NAD about twofold. That still leaves the NAD concentration in the fed state well below the Km for either SIRT3 or SIRT1. So our approach is to combine these NAD donors with leucine, fairly high concentration of leucine. And when we do that, as you can see on the right-hand part of the slide, we can show an increase in lifespan in preclinical models, an increase in fat oxidation, insulin sensitivity. In a mouse model of atherosclerosis, the LDL receptor knockout mouse, we can show a reduction in atherosclerotic plaque size, a reduction in macrophage infiltration. And importantly, these are done at concentrations where we see no independent effects of leucine, nicotinamide riboside, or nicotinamide mononucleotide. So we believe we have a means of enhancing the efficacy of excuse me, enhancing the efficacy of NAD precursor, NAD donors. However, we do not yet have clinical data to support that. These are all from non-clinical models. So if you go forward one slide, we'll take a different approach. Resveratrol has been a molecule that, lost count of the number of papers published on it years ago. Again, a molecule that has shown high hopes, has tremendous effects in non-clinical models, primarily as a sirtuin activator. However, the concentrations required are difficult to obtain. We have bioavailability problems, essentially due to rapid metabolism of resveratrol. So we asked the question, could we make the achievable serum levels of resveratrol functional by adding leucine? And what we've done here, this is a quick summary, but what we've done here is we have treated DIO mice with varying concentrations of resveratrol with or without leucine. And when we do this, the resveratrol gives us nothing. We are simply, unless we push the resveratrol to very high concentrations, we are unable to get an effect. However, when we combine it with leucine, what you can see in what I consider to be two dramatic photographs, you see PET scans of a control mouse in the upper left, and in a leucine resveratrol treated mouse in the lower left, and you can see the FDG lighting up enormously. In fact, we have a 60% increase in FDG uptake, reduced plasma insulin and HOMA-IR by more than 50%, comparable reductions in inflammatory biomarkers, and along the way, a salutary effect on body weight and adiposity. And again, this is at concentrations that have, that we cannot, this is at concentrations that show no independent effects when not put together in combination. The lower right part of the slide simply shows you that in a single clinical trial in pre-diabetics, we were able to improve glucose tolerance, insulin response to a glucose load. HOMA is shown because that's all I had room for in the figure. So this does translate clinically, although we've not taken it beyond pre-diabetes yet. Can we go to the next slide? So we've also asked the question of whether we can generate a pharmacological approach. And what we've done here is we've put together a combination which we call NSO-200, for which we have an IND that's been through a couple of phase 2 trials, in which we are combining leucine, low-dose metformin, the starting dose of 500 milligrams, and an extremely low dose of sildenafil, 1 milligram. Now, the point of the sildenafil is not PDE5 inhibition. At very low doses of sildenafil really do not produce appreciable PDE5 inhibition. Instead, it is for activation of eNOS, which then interacts downstream with both SIRT1 and AMPK in a positive feedback loop. Let's move to the next slide. And so when we've done clinical trials in both non-alcoholic fatty liver disease and in obesity, in the upper left, what you'll see is liver fat, and you see a decrease in liver fat with NSO-200 in individuals who have some evidence of liver disease. Full disclosure, this is a study for which the primary endpoint was not met. That primary endpoint was drop in liver fat in all comers. It turned out to only decrease the liver fat in individuals who had ALTs over 50, which was half of the cohort, so some evidence of liver disease. But even in that study, we showed remarkable decreases in blood pressure, in triglycerides, improvement in insulin sensitivity, and reductions in weight, which led us to take this forward in a separate IND for obesity. And you see the full obesity cohort on the lower left. and you can see a nice time-dependent, follow the purple line, a nice time-dependent decrement in body weight. This is in the absence of any lifestyle modification. There were subcohorts, such as African Americans, that had close to a 6% decrease in body weight. But the overall group had about a 2.25% decrease in body weight. Again, that is without any lifestyle intervention. We believe that with lifestyle intervention, which is the normal way you would run an obesity clinical trial in phase 3, we will achieve a much larger synergistic with lifestyle intervention decrement in weight. Let us move forward to the next slide. And I'm going to end on this slide because I'm well aware that leucine presents us with a paradox. And the paradox is leucine is considered to be an activator of mTOR. And so why do we see the effects we see if you're supposed to be activating mTOR? And the answer is the concentrations that are required to activate mTOR are fairly high, in excess of 1 millimolar. The doses that we give get us to about 0.5 millimolar. It's really hard to get to 1 millimolar. Fasting levels are around 0.1 millimolar. Levels following a good high protein meal is between 0.25 and 0.3 millimolar. We raise it to about 0.5 millimolar, and that is sufficient to activate SIRT1 and SIRT3 without having any effects on mTOR, and we have measured this. So then how do we explain the observation that it is also a marker for metabolic syndrome in some studies? And the answer there is that when you look at the enzymes responsible for metabolism of leucine and the other branched chain amino acids, and especially focusing on branched chain keto acid dehydrogenase, what you see is there is a block there in insulin resistant states. And it is the buildup of that metabolite, ketoisocaproic acid, that really seems to have the effects on mitochondrial dysfunction and oxidative stress. However, higher concentrations of leucine have been shown to activate branched chain keto acid dehydrogenase. So we believe this is the resolution to the paradox. But what we can say is clinically, we see the effects that I've just presented. And I think I've run out of time, and I've been warned not to go over time. So I will stop there.
Thank you, Michael, and the whole panel. First sound check. Can you hear me?
Yes.
Super. You know, those are great presentations. We've gone a little bit long with these highly informative presentations and with some great questions. Doug, would you like to moderate a short discussion within the panel based on things that you've been hearing from the other speakers for like 1 or 2 driving questions that are motivating the panelists about the other panelists' presentations?
Yes, thank you. I think one of the questions that came up early on, and also one that I had made a list of before the meeting, was, so what is the relationship between mitochondrial function and stem cells and postmitotic cells in relation to aging? And maybe I could have one or more of the panelists give their thoughts about that. Okay, well, I will mention a very interesting study from Niels Larsen's lab. They made a mouse where they inactivated the proofreading function of mitochondrial DNA polymerase gamma, and that increases then mitochondrial DNA somatic mutation rate. And these animals prematurely age. And one of the interesting early studies suggested that much of the premature aging was the accumulation of mitochondrial DNA mutations in stem cells that then, in fact, became distributed in the postmitotic cells. So I think that both are relevant. Then the question is, how do stem cells keep their pluripotency or their potency over lifespan? I don't think we really have an answer to that, but we do know that many of the stem cells are basically glycolytic. So maybe, in fact, they're using metabolism to protect the mitochondrial DNA. And it is well known that if you look at the stem cells in intestinal crypts, that you're accumulating these mutations in these stem cells at the bottom of the at the bottom of these crypts. And then once a mutation occurs, it becomes increased in heteroplasmy level as the cells progress up the crypt, and that seems to be related to the incidence of aging. So I think that there is a lot of interest in the relationship between stem cells and postmitotic cells, and they may in fact have quite different effects on the phenotypes of aging and health span. So I want to ask a question just in general of the panel. What is health span? You want to talk about that? Oh, I'm sorry, Francesca's going to comment.
Just because I wanted to tell to that point, because what's very interesting about the stem cells, also the correlation in cells like oocytes, which they have to maintain a level of fertility over the lifespan, and the health span of a woman, which is about until 40 years old. And so what's very interesting is that one of the pioneer of mitochondrial metabolism studies, Otto Warburg, started his research by looking at fertilized eggs that would have spikes of ROS upon fertilization. So his idea was that all the oxygen was all of a sudden consumed upon fertilization, but the reality was that the mitochondria was switching from one metabolic state to a different metabolic state, which is the same as it happens in in immune system for T cells. So your circulating T cells, they rely mostly on oxidative phosphorylation, and then upon infection, they switch to they have a metabolic switch where they start over proliferating, and of the like similar to cancer, which is switching from OXPHOS to mostly glycolysis, which is also what Warburg was saying, and this is how your PET scan is done. It much more increase in glucose utilization. So all these different things says a lot about what is the metabolic state of the cells, what determines the metabolic states of the cells. And it's the same as there's a parallel between stem cells, immune system, oocytes, fertilization, and probably many other different things. So everybody was asking the question now, there's like big thing out there about metformin used during bouts of exercise. Is it good or not to use metformin on top of exercise? So the question there is, what if, during a bout of exercise, your skeletal muscle switch from some metabolic states to a different one? And what if metformin is not good at that particular stage? As I show in my presentation, for instance, an uncoupling current, which will actually help this switch, is not supposed to be there all the time. It's supposed to be there only a specific moment in time. It can be triggered by ischemia. It can be triggered by temporarily metabolic, switch. And so is it good to go after, intervention, for instance, with metformin when we're actually healthy and there's nothing wrong with us, and we can fix things that are not damaged as yet? And so what probably near Barzilai and everybody's asking, do we have, or FDA for that matter, do we have actually very good endpoints that can determine at what point, at what stage, at what point in time are we supposed to start taking metformin to prevent aging? So for us in our company, we're being very careful. We're just going after disease and maybe go back a little further to see how, along the way things change. But again, this metabolic switch, as you mentioned, for stem cells is very important for many other tissues in our body. Your muscle is fermenting when you're working out. Your cells, your T cells are over proliferating and using glucose when you encounter an infection. Your cells become over proliferating and become cancerous when the Warburg is not stopped, and it becomes a chronic exposure to a different metabolic switch that the cells are not supposed to be under, and so on. So sorry for interjection. I just had this thought, and I thought it was
that's great.
Mentioning.
Thomas, do you want us to go on with panel discussion, or would you like to look at some of the chat questions? Because there were quite a few interesting ones that were coming up. You're muted. You're muted, Thomas.
No, you're
okay.
muted.
I couldn't read that sign language, but I wonder if we could continue this metformin conversation for another moment.
Oh yes, go ahead.
Just thinking about what Francesca has just said, it's true. many of our colleagues, I'm sure many people in this webinar are taking metformin for off-label indication, or health span related indication. And the presumption is that it is harmless at least and helpful at best. And certainly there is an abundance of evidence for its helpfulness, but to engage in a broad population recommendation for taking a drug like metformin, frankly, concerns me a little bit without a study that specifically, like the study NEAR is doing. When that study is complete, when we have tame data to say that, yes, we have compressed disease span and expanded health span, I think it may be time for me to start taking metformin. Until then, the assumption that, as they say, couldn't hurt, I think there have been too many small molecules that couldn't hurt until they did.
Great. So obviously, for any clinical intervention, we need both a safety and an efficacy study. And that's, of course, why we have an FDA and why they are so careful with these kinds of things. So thank you very much for that comment, Michael. Let's see, trying to find some interesting questions.
Doug, can you hear me?
Yes, now we can hear you. Perfect.
Excellent, thank you. Yeah, you were asking before whether to continue with intra-panel discussions. We found that the audience questions get shortchanged in many of these, so I was going to say let's go do it. And I think EJ has posted all of the questions or some of the prior questions in but it's downloaded. So let's see if we can put it all in the front. But I do remember a question from Michael Fossil, who's been posting some of his questions in the chat box. So if the panel could take a look at some of these and just sort of a toss-up, jump ball, pick up something and go for it, we'd be very curious.
So they're at the top of the list. I'm sorry. So
it's just in the most recent at the bottom of the chat column. I mean, I can read a couple things here.
I found one maybe for Michael that I can read to him if Doug doesn't see it. So Anna Madalena asked Michael, how long does ND 0 to 100 stay in the body before being eliminated? How well is absorbed? Also, when will NS 0 to 100 be commercially available and in which countries?
Wow. So the answer to the last question is easy. Right now, nowhere. It has to go through. It's only been through phase 2. It would have to go through phase 3 trials and ultimately an NDA before it's available. Before availability can be discussed. The duration, right? It is twice daily dosing, BID dosing. The half-lives of all 3 components, metformin, leucine, and sildenafil, are rather brief. The biological half-life of leucine is a little bit longer, not the PK half-life, but the pharmacodynamic half-life because once you have activated the sirtuins, their effect down the cascade is longer lived, but even so, it is hours, not days.
Doug, maybe I can read up. I'm sorry, go ahead, Doug.
No, go ahead, Michael. Thomas.
I was going to read a couple of questions from Michael Fossil and from Steve Engel and from Claude Lardinois or Lardinois. Michael Fossil is asking if mitochondrial aging drives aging, why does this not occur in the germ cell line? That is, if the mitochondrial lineage is more than 1 billion years in temporal extent, why do mitochondria only show aging changes in somatic cells after fertilization?
May I answer that as a start? This is a topic of great interest to us. So as Francesca said, we have really what's called the situation where the egg starts out with a single cell with about 1 million mitochondria. It gets fertilized. The cell undergoes division to give the blastocyst without any mitochondrial replication. Then only a few of those blastocyst cells in the inner cell mass, just a small set of those cells, give rise to the next female germline. And the number of mitochondria in those primordial germ cells has been actively studied. In humans, it's been estimated that it's about 4. It can be up as high as maybe 100 in mouse. Then those primordial germ cells replicate, and they ultimately give rise to the cells that are then going to differentiate into the oocytes. And so those mitochondrial DNAs that get amplified up to 1 million, and then at fertilization, they go through the cleavage process and go back through a bottleneck. So what you're doing is you're getting then a high mutation rate, but with severe bottlenecking. And the bottlenecking then means that you're going to generate, if you have a majority of wild type mitochondrial DNAs, then most of your oocytes are going to have the normal mitochondrial DNA. When you reach into, say, if you think of a bottle of red and white balls, and you reach in and grab only 4 balls, and let's say there are only 10% are red, then most of the time you're going to get white balls, and then your next oocyte's going to be genomically normal. But every now and then, by chance, you're going to reach in and get a mutant. And the mutant, if it's very severe, gives you maternally inherited disease. And then selection immediately eliminates those. So the reason mitochondrial DNA disease is so common is because, in fact, of the sampling situation. So then you're constantly going back to a germline, a normal type, by this population bottleneck. What's interesting about that is if you get a variant that might be mildly beneficial, then that can also get sampled and go and rapidly form to a pure mutant germline. And if that's beneficial in an environment, then that then rapidly becomes amplified in the population. And you then have this regional specific mitochondrial variants that we see that are adaptive to these different environments. So that's a very good question. And it really, we believe, very important in the evolution, intraspecific evolution to environmental change.
I think that's a pretty comprehensive answer. And by the way, if there's any follow-up questions, people please feel free to, I'm sure that our panelists will be happy to engage in more detailed discussions with you on these questions. There's a question from Steve Engel, who's CEO of Cobar. Obviously, we presented the interest of several companies here, but there are many companies targeting mitochondria. And Steve asked the question, any views on Dr. Gilbert Ling's work on the importance of ATP versus the induction of proteins in intracellular water using movement of electrons?
Thomas, I hardly heard you, so maybe if I could read the question.
Please, yeah.
No, I have to find it though.
Can you hear me any better now? I'm holding the mic to my mouth.
Yes.
Yeah.
So Steve's question is, any view on Dr. Gilbert Ling's work on the importance of ATP versus the induction of proteins in intracellular water using movement of electrons?
I cannot speak to that. I'm sorry, I don't know that work. I don't know, Doug. Sorry, I'm not familiar with this work, but send over the paper. Maybe we'll look into that and have this conversation.
Yeah, Steve, maybe we can put you together with Francesca and the panel, and you can have an extended discussion. Sorry, go ahead, Doug.
Well, I don't know if this is what the question was about, but there's a lot of interest in the structure of water around molecules, protein molecules, and especially around the ATP synthase. And there's also great interest in using near-infrared red radiation to change the structure of water around proteins. And there's data that suggests that the ATP synthase, which is basically a spinning wheel that drives the ATP synthesis, can be changed by near-infrared radiation, thus changing the structure of water. And there's also growing interest in whether near-infrared radiation can actually affect the efficiency of electron transport chain and maybe even decrease ROS production. So there's actually quite a few interesting companies that are looking at radiation as a way of regulating bioenergetics. So I think that's an interesting area, not one we had represented here today, but we could have.
So here's a general comment from Claude Ladouanis. Insulin resistance, not metabolic syndrome, is associated with a lot of chronic disease. I wonder if there's been any studies looking at a link between insulin resistance and mitochondrial dysfunction. I would hypothesize a strong link, especially with increase to free fatty acids seen in insulin resistance.
So that's a very difficult question, at least for me. I'm not a clinical physiologist. I can only speak to and quote people that I heard having this conversation. All I can say based on my experience, particularly in the biophysics of this and reading on papers or reviews on mitochondrial physiologists. One of them is David Nichols at the Buck Institute, where was trying to discuss, the release of insulin from beta cell in the pancreas, which is very difficult to study, by the way. It's not the low-hanging fruit, and this is why we still do not know very much about mitochondria in the pancreatic beta cells. But for instance, it is kind of known that it is ATP made by mitochondria that drives insulin release in beta cells. So that goes in circles again with the conversation if is the egg before the chicken or the chicken in the eggs, because the question is, it's insulin dysregulation that causes insulin resistance, or there's a first issue before that. So I would have my personal bias in considering, as Ralph DeFronzo put it, which by the way is one of the scientific advisory board at Pano, Ralph DeFronzo, which is a clinical physiologist that studies insulin resistance and diabetes for 45 years, has always has one of his favorite thing that he always argued with FDA is that, in his opinion, that before there is insulin resistance, there is insulin production issues at the level of the pancreas, so which causes, circulating insulins, which is higher, and so on and on. So that's all the little things and bits that I could get from clinical physiologists, but and of course my personal bias would be that is the mitochondria to drive insulin release from the pancreas that over time, for instance, a redox stress like fuel oversupply, like you're obese and you started to get diabetes, ROS would actually greatly increase and switch pancreatic beta cells from one type of metabolism to another, which basically is saying that you don't make ATP anymore, insulin is not released as efficiently, and so on and on and on. But it's such a big question that I only can address it on my little piece of expertise in the biophysics of mitochondria for that.
Yeah, this is Claude Lardenois. Can I just make one comment? I think we should really get rid of this metabolic syndrome thing, because when I work with Jerry Reven, who's the father of insulin resistance, we had many patients that were profoundly insulin resistant that didn't meet the metabolic syndrome criteria. And it's kind of a hodgepodge of things tossed together. And the real driver is the insulin resistance, because that's what's going to raise the blood pressure, cause the dyslipidemia, and all the chronic disease processes. So I would encourage people to use the word insulin resistance rather than metabolic syndrome.
Right. I actually was listening to one of Tia's podcasts about 2 weeks ago with Gerald Schulman, and they were stressing the fact that even healthy individuals, Tia was studying, or Gerald Schulman was studying young college students in their 20s, and they are, some of them, even healthy and exercising, have insulin, their insulin levels are high and they're insulin resistant. So I don't know if that can be described as part of this issue, but that's why I think it's a very difficult conversation, and everybody has their own favorite things. I know Ralph DeFronzo has his opinion, but yeah, that's
Michael, you're leaning forward.
Claude again. This is Claude.
Yeah,
I want to comment
Claude.
on Francis's. Yes, I happened to listen to that thing by Gerald. And I think what the real take-home message there that people forget is that there are more people that are insulin resistant that aren't obese than people that are obese. And we kind of get locked in. You have to be fat to be insulin resistant, and that's totally not the case. And he showed in college students, 10% of them had glucose or had glycogen synthetase defects in the muscle. And these people were lean, healthy, and everything else. So let's toss out this thing about obesity synonymous with insulin resistance. They're not the same. There are several obese people that do not have insulin resistance.
And
yeah, maybe Hazel can comment. Hazel?
Yeah, if I may add to this confusion in all the work that we have done with the peptides by lots of people, we have found that the biomarkers that we normally measure, blood glucose, blood pressure, cholesterol levels, body weight, the peptides don't seem to affect any of those, and yet they protect the end organs from the damage. So in other words, you can have retained high blood pressure and yet not suffer the renal and cardiac consequences of that. You can be diabetic, blood glucose doesn't reduce with the peptides, but yet they protect the kidney, the retina, and the heart. So I think that we got to separate a little bit from what we can easily measure from where the real pathology is. So the pathology is looking at the end organ level and not as what is circulating around. But I don't
Michael,
have an answer to this either.
I just wondered if Michael wanted to comment too. He hasn't had a chance to talk.
Well, only that first, I agree with Claude's insulin resistant centric view of this universe. But beyond that, to address his question, in terms of the relationship between insulin resistance and mitochondrial function clinically, I don't know if we can address the relationship with mitochondrial function clinically, but we can address the relationship with mitochondrial density clinically. And I'd have to go back to find the papers. They're more than 10 years old, but there have been papers that have looked along the train of insulin resistance from highly insulin sensitive to highly insulin resistance, and looked at via biopsy mitochondrial density in both adipose tissue and skeletal muscle. And there you do see an inverse correlation, higher resistance, lower density. But that's as close as I can come to really answering the question. That's associative, not cause and effect.
Well, we did one experiment that was kind of interesting. We created a mouse in which we put a single point mutation in the cytochrome oxidase subunit 1 gene in the mitochondrial DNA. Mm-hmm. And that animal develops a glucose intolerance and insulin resistance. So that then says that if you go directly to the mitochondria, you can generate the systemic defect without any of the other overt problems. So just another possible prediction. Thomas, another question?
Yes, I'm looking at Christine Peterson. Since this is an electrostatic process, could EMF possibly disturb the process?
Anybody want to venture there?
I cannot hear Thomas very well. I wish you can someone else read the.
Sorry, yeah, I think I have to hold this up. Go ahead, Doug.
No, go ahead, Thomas. Just say it again, please.
Yeah.
Oh,
I'm trying to.
I have to go back up
in the chat. I can't.
Now, here the basic question was: if it's an electrostatic process, could electromagnetic fields interfere with the electrostatic system and therefore energy production? I think that's a very exciting and hot topic. So, what are the panelists thinking about it?
It's an amazing question. I don't know how to address that. I cannot speak to that specifically. I just know that I talked to a biophysicist that works in NASA, Yuri Griko, and they're working on that because they're actually basically studying what is the effect of electromagnetic field on astronauts when they go to space. And they have a rodent model that they use because they have chambers that they can use to expose the mice to the same conditions as humans across the space. And one of these parameters is actually electromagnetic field. And he has a paper on that. I can send it over to you. And so they're thoroughly investigated that at NASA for now. That's all I can say. I can't elaborate further, but sure, there is potential there to affect.
Hazel or Michael, did you want to comment?
I really don't know anything about this to say. I have nothing to contribute.
Yeah, well, we're keenly interested in this question, and we have actually a whole program called quantum bioenergetics, and really interested in how the biophysical process might be interacting not only with the environment, but how the process itself might be a signaling system. So one of the things we collaborated with Francesca on is that the adenine nucleotide translocator, which is found in almost all cells, is a voltage-dependent proton channel. So the idea is, as the electron transport chain pumps up the proton gradient, when it reaches the electrostatic potential that's too high, it opens the channel and then relieves the pressure. But if you do that, then you create an oscillating system. And by the classic equations, an oscillating electrical field will give you an electromagnetic field. So we're interested in whether, in fact, there is a signaling system via electromagnetic radiation. But that's all very speculative, but it's certainly an interesting idea.
There's another question from Michael Fossil. By the way, Michael made some favorable comments about my mitochondria virtual background. I stole the idea from Leah Barzilay. I love your background, Michael. Michael, your second question was, how can we characterize the functional differences between mDNA and nDNA sources for the mitochondrial aerobic enzymes? Is there a way to predict which enzymes are mitochondrial versus nuclear in origin?
Actually, let me clarify that if I might, Thomas.
Yeah,
what I'm
please.
really getting at is the function of them. And it seems to me that most of the enzymes that are coded in the mitochondria need a rapid response time to any aerobic change, whereas those that are coded for in the nucleus tend to be more structural elements, and there's a little more lag time and a little more time for temporal slop in response. I'm not sure how to put that any better, but you know what I mean.
Yeah.
What about our panelists?
So, well, I don't even know where to start, but actually Doug also has amazing papers. The last two papers that Doug came out with was about combination of nuclear genes with mitochondrial genes, how do they affect bioenergetics and energetic metabolism. So the combination of your nuclear gene will affect, we are a chimera of nuclear gene and mitochondrial genes. So the ones that you immediately have to modulate super fast are the ones which are in your mitochondrial DNA, the 13 polypeptides. So those have to be modulated by very quick signaling molecules, which are like ROS, ions, oxygen sensing, all these different things. And then everything else should trigger a retrograde signaling pathway that goes back to your nucleus, and over time makes changes such that your cell chronically change, and they acquire a phenotype that allows them to support and stand those stresses. So for instance, if we're talking about obese people or people with diabetes, think about fuel oversupply. You eat too much. Your electron transport chain is oversaturated all the time. You have a hyperpolarized potential all the time. If there was not a backup plan to save the mitochondria for that hyperpolarized potential, they would be dead. Instead, they're obese and they're hanging in there, but they're actually doing this. Their cells, they're surviving the stress, but they're getting exposed to an oxidative chronic stress that actually is toxic to them. So that's the difference between, and so from an immediate reaction, acute response to the stress, it becomes a chronic response. So this is the big, large question. Are nuclear genetic mutation the drivers for mitochondrial mutation and then metabolic switch? Or is the metabolic reorganization of the cells that triggers nuclear mutation that drives the phenotype of disease conditions, pathological conditions? So if you go to cancer, we have, what's interesting that there used to be a time when we're talking about individualized medicine. But the point is that if you're going to the hallmarks of cancer and you take any cancer, there's a tissue agnostic phenotype of all of your cancers. That is, they eat glucose more than, so they're more fermenting than oxphos, and they have a bunch of nuclear mutation which are expressed across different tumor. And this is called the basket trials that now FDA is going after, which is a tissue agnostic way of looking into tumors. It's not, there's like drivers mutation that you could find across cancers. How come all of this mutation are common across different type of tumors? And so the big question is it something that comes before that, which is the metabolic switch that drives retrograde signaling that drives your nuclear gene. So that's the long term as a reaction to the short term. But that's a very simply.
As a simple-minded professor of medicine, I sort of look at it as if I wanted to change the proteins in the mitochondria, if I hold my breath, it's going to change the ones that are operating right now in the mitochondria. Whereas if I change my diet or use thyroid, I'm going to be changing the nuclear transmission.
And sure.
Well, Hazel or Michael, did you also want to chime in on this interesting question?
This is a fascinating question. I rather liked the way Michael Fossil just put it in terms of rapid response versus prolonged response. I wouldn't recommend that breath hold for too long, but beyond that, I don't have much to offer.
Yeah, but I'll give you an example, to Michael, like ischemia reperfusion. You have an immediate, you have a short or long, whatever, exposure to ischemic insults, your heart, your brain, and everything, right? So you need to have a backup plan, which is immediate to that. And then reperfusion is basically over time, which is what actually basically kills us. And we still don't have a solution for that. You go to the hospital, you try to resolve your ischemic insults with oxygen reperfusion. But that drives over time a chronic exposure to ROS, which your body's not used to. And so that you have an acute response that then translates into an immediate response. But that acute response is your mitochondria. If you didn't have a backup plan, which is an acute response to that insult, you ain't gonna live. You're You're just gonna die. So yeah.
Well, I was kind of asking a teleological question or an evolutionary question, which is if you're going to pick which genes to put in the mitochondria and which to put in the nucleus, how would you divide them? And I think that's where Michael's about to go too. Go ahead, Michael.
Or Doug, maybe.
Or Doug, I think was.
Hi, Doug.
I was just going to say the beauty of Michael's question, and then I'll let the other Michael answer it, is why do we have a mitochondrial DNA at all? I mean, if we were transferring, 2,000 genes to the nucleus, why do we keep 13? I think one of the interesting things that is possible is by changing the mitochondrial DNA copy number, you can have a tremendous physiological response on a very short period of time. Likewise, by changing heteroplasmy levels, you can change without acquiring new mutations. You can change physiology to a dramatic effect. But then I'll pass it on to Michael Zemmel.
Well, Doug, I appreciate that, but I don't know that there's anything I can add to that. Again, as Michael said, we're dealing with a teleological question. This sure, it makes sense, but how do we know? I'm sorry, I have nothing.
It's a fun question, but we can't answer it, right?
Well, no, maybe, I think they're really good answers, but maybe we can go on to some other question. Michael Foss, let's get back to you about that. I'd be delighted to talk to you more about it.
Terrific. This is the question. Yes. Can you hear me?
Yeah.
Yes.
Okay. This is a question for Hazel. I think Claire Stewart asked, given the potent role of SS31, is there an increased risk of tumor formation?
Yeah, well, that's a fascinating question. And that was something that really dogged me at the beginning of the studies. I was very worried about that. But in fact, we have now done a number of studies with collaborators on cancer cachexia. And it turns out that the SS31 will protect, the skeletal muscles, the heart, and the diaphragm from cancer. But the tumor size itself does not change. So it doesn't have anything to do with increasing tumor size. Now, that doesn't address the question of really tumorigenesis, and that's something I don't know at this point.
Okay. I just lost my place here. Daniel Adekunbi asked, this may be unrelated, sorry if that's right, but please, how can I selectively repress glucocorticoid receptor translocation? Sorry, my eyeglasses are not focusing on the letters, to the mitochondria in primary cell lines without altering nuclear GR signaling. Any thoughts on applicable small molecules or vector transfection? Thank you. Seems like Daniel has a very specific problem he's looking at. Can we help him?
Oh, wow.
I don't know of anything.
Maybe
So,
if anything occurred, so go ahead, Doug.
so the question is, glucocorticoid receptors internalized and turned over. Was that the sense? I wasn't quite sure.
Daniel, are you still here? Do you want to unmute yourself and clarify or amplify your question?
Yes, I'm here. Thank you for this privilege to get this technical question. I'm basically interested in differentiating G-house signaling in the mitochondria from the nuclear G-house signaling. So an idea that came to my mind is to stop one organelle G-house signaling, and then the other I can study effectively. So that's why I'm wondering if there is a molecule or any transfection technique that I can use to inhibit the translocation of glucocorticoid receptor into the mitochondria. I will so much appreciate that. That's just a question here.
Well, the similar experiment, but not that one, we were very interested in the fact that the estrogen receptor beta is located in the mitochondria, about 20%. And that's really interesting because it's activated by estradiol. So if you add estradiol to either isolated mitochondria or to cells, within 60 minutes, you'll double the antioxidant defenses of the mitochondria. So there are bifunctional purposes for having mitochondrial, plasma membrane, and nuclear receptors. So what we did to prove that is we knocked out the resident receptor, and then we created receptors where we had made point mutations that targeted the receptors to the different organelles, and then used each one and then looked at the physiological outcome variable. So if you're asking a technical or experimental question, that was for a similar experiment, that's how we addressed it. But if that's not your question, I apologize.
So if the panelists have any further ideas, happy to put you in touch, pass them through to Daniel. Christy Carter asked a question. It's interesting that leptin is not included in any of these mechanistic models. I think this is Michael Zemmel's slides, and is a major CNS regulator of food intake and metabolism. Any comments?
So mechanistically, leptin simply does not fit in this pathway. Obviously, you're talking about CNS regulation of hunger, appetite. Obviously, leptin has a number of peripheral effects as well, but they don't really fit in this sirtuin pathway. I will simply say that passive measurement of leptin is of interest, and that passive measurement does track the other clinical outcomes and preclinical outcomes that I described, meaning when we have successfully activated sirtuins, we do see not only a decline in adiposity, but a corresponding decline in circulating leptin. But again, the signaling pathways we're talking about don't cross, at least not to the best of my knowledge.
Hazel, did you have a thought? Francesca?
No, it's not my space, but I think Michael is perfect for these kind of answers.
Okay.
It's not my space, and it would be up to Michael as far as I'm concerned. So, yeah, no.
Okay, we have to leave leptin.
Okay, sorry. Ebenezer, Ajayi asks, can the endoplasmic reticulum help the mitochondrion in the mop-up of excess assaults naturally occurring in the electron transport system?
Yes,
Excess of salt.
Repeat that, yeah.
Mop up the excess assaults naturally occurring in the electron transport system. Can the ER help the mitochondrion?
Well, the ER can certainly help by initiating mitophagy if there are damaged mitochondria. So generally, mitochondria will have to undergo fission first They become smaller and then they get wrapped around. And we see this actually in many animal models of chronic diseases like diabetic kidney disease or ischemic injury. You will see evidence of small damaged mitochondria inside large autophagosomes. So that's how the ER can help to remove damaged mitochondria.
We have a question from Charlie Alexander. There are people with insulin resistance who do not have obesity and people with obesity who do not have insulin resistance. HOMA-IR is not a good way to find people with insulin resistance because it's a test that uses fasting insulin and glucose. A more sensitive test for insulin resistance requires measurement of postprandial insulin and glucose or a glucose clamp.
Mm-hmm.
And
those were comments, not questions.
Right. Yeah.
Yeah, that's right.
true.
Todd Lawrence had a question. How does fasting glucose compare to HOMA-IR in terms of both positive and negative predictive value, assuming that a glucose clamp is the gold standard related?
And that was followed up, I see, by another comment that says fasting insulin almost as good as HOMA-IR. Fasting glucose depends more on beta cell function than insulin resistance and is not a good measure of insulin resistance. And of course it's not. But the start of this thread is, how do you measure insulin resistance given that you've got this imperfect tool of HOMA, which is the low-hanging fruit, but is not fantastic, and you're not going to run glucose clamps on a population.
Mm-hmm.
Well, this is Claude Lardnois. Let me chime in a little bit. I've looked at this insulin resistant thing for a lot since this is my whole life's blood. And you're right. I think the biggest mistake the ADA and everybody's making is they're relying on a fasting value. They're not challenging the beta cell. And so I think what I do in my practice is I do a 75-gram glucose load. I measure the 1 hour and the 2 hour. If the 1 hour is greater than 125, and the 2 hour is greater than 120, I concur that person has insulin resistance. And like you said, you have to do the clamp, which is the gold standard. But when we tried to analyze this at Stanford, those two numbers look pretty good for predicting who is be insulin resistant.
Are you capturing insulin at the same time points, and does that help your prediction?
We did in the beginning, but in clinical practice, I'm not doing it because I think it's hard to interpret. I think clearly if the glucoses are high, it's telling you that there's inadequate insulin.
Right. I was asking the question to differentiate between inadequate insulin and insulin resistance.
Oh, yeah. Okay. Yeah, you'd have to do the insulin levels then because of that new heterogeneity of diabetes that they've been talking about. Good point.
So this can go on for a long time. Fascinated to listen to this. We do have another conference or starting on this line in a few minutes. So a couple of announcements. Again, we're going to have a recording of this call or a Zoom conference available. All registrants will get links to it automatically. It probably will come out in the next several business days. We also will make the slides available. I think if we do what we've done in the past, we'll also try to get it transcribed, although that might take a week or two. And we'll also reproduce the chat so people can, if you see something interesting going on amongst the chatters, you can reach out and talk to them. So I would like to thank everyone, the panelists particularly, and the attendees for coming, and wish you all a great weekend. I would bang the gavel and call the formal portion of this this webinar done for now. People are invited to mill around, and if you want to open up your mute and give shouts out to folks, please feel free. We will be closing the room in about 5 minutes. Thank you very much.
Thank you, Thomas.
Thank you all. Thank you.
If I may, I just want to say something for the record because many people did leave a few minutes ago, and I want this to be in the chat. We had 1,000 folks joining us in October for the MetaBesity conference. We expect that we'll have about 1,500 or more in October 2021. And when you listen to this panel, you can see why. And what's so important is that many of those people were on for 4 days and they were not gerontologists. What is happening here is just amazing. The ability to focus on prevention, not just cure, and to get this information out to all folks in the professions of aging is really catching on. So I wanted to thank everybody very much and also make the pitch that if any of you who are reading this chat or are still on, get an email from the Catalyst Institute, make sure it doesn't go into trash or spam, because it's going to be an invitation for some exciting webinars for our October meeting, which is a huge annual meeting, but also for you to participate. And we want participation from all the stakeholders. And that's growing, particularly because of COVID and because the United Nations has named the next 10 years as the 10 years and decade of longevity. It's a big deal. Thank you.
Thank you for that, Adrian. We look forward to collaborating with you all. The more we have these kinds of get-togethers, I think the more exchange of ideas and questions, and very much looking forward to that. So thanks very much. Have a great weekend. Bye-bye for now.
Thank you.
And thank you, panelists. It was great.